Dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me?
Thank you, David. Thank you, everyone. Está apagado esto, ¿no? Sí, perfect. So thanks for the introduction. Thanks for receiving me. It's been already 3 times in the last year. If you keep inviting me, I will keep coming, just to let you know that. But this time I'm presenting what I'm doing in my lab, which is basically how to implement system biology approaches to understand how fruits ripe, how fruits accumulate secondary or specialized metabolites, specifically non-climacteric fruits. But before I go into detail what we do in the lab, I need to do some advertisement of my institute. This is a very recently established institute in Valencia. It's a joint center between the CSIC and the University of Valencia. We started 2016 with the first groups from the university establishing there, and since then a lot of young people have been establishing their groups. Me, although I don't consider myself young anymore, but yeah, it's a very nice environment for young researchers to start developing their interest, especially in our main topic in the institute, which is computational biology. So it's a fancy, a long name, Institute of Integrative Systems Biology, even more complicated acronym, I2SysBio. I just call it I2. And it stands for different researchers working in computational biology, trying to adapt different methodologies to understand living organisms. So it's not only plants, it's all types of organisms. In the institute, we have people working in human viruses, in any type of pathogen, in plants, in yeast, bacteria, and so on. The institute has 3 main programs. They have specific tasks and research lines, but there are also transversal research actions. So the different groups, we are collaborating with each other to follow up a particular research action. And the research action, all of them revolve around the notion of the One Health concept. So we are all interested in improving the health of ecosystems, of humans, of plants, and we have different actions that allows us to collaborate. And this is just a very interesting thing. A couple of years ago, we decided to restructure our institute and create new departments, and this is actually something very healthy that I would recommend all institutes to do it. We basically did a scoring of different terms among all the PIs of the institute, and then we simply generated a PCA based on the scores of those terms. And you can see that there are clear separations on the PCA, and this was like the main idea behind restructuring the institute. So now, because of this, we were able to allow ourselves to divide in 3 main groups. So these are the 3 main programs. The program in which I am right now the coordinator, the director, is the Systems Biotech program. And as you can see here, one of the first terms that gives more score into this clustering is metabolism. So that's exactly what my group does. What we are doing in my lab is trying to understand how plants regulate the production of metabolites. So my group is called, also a long fancy name, Transcriptional Orchestration of Metabolism. Orchestration because we're interested in the main regulators that regulate other regulators. And these are basically all the different types of methodologies that we implement to try to understand how plants regulate metabolism. So we do large TF family characterization using both bioinformatic and molecular approaches. We do different types of omics. So we sequence genomes, we annotate genomes, we do metabolomics, transcriptomics, proteomics, and so on, and we integrate all of those omics. We work a lot with network theory. That is one of the things that I'm going to present today. We also try to study different plants, and we try to identify natural variation genotypes and try to understand them from the omics point of view to identify new regulators. And we are interested in this case with secondary metabolism and development. We are also using a lot of our efforts to develop cell suspension models like the ones you see here. So we are moving from plants to cell suspensions because it's quite easy to stimulate metabolism in these systems. And we finally develop databases and visualization platforms that, if I have time, I will dedicate a little bit of time today for that. We have different species of interest and different study models. We are interested in species that are for the food industry or for the pharmaceutical or cosmetic industry. So you have some examples here. We work in grapevine, in tomato, in cannabis, and other species. So why we are interested in the regulation of plant specialized metabolism? Because it's from the basic point of view, it's a pathway that produces compounds that are very necessary for the interaction with the environment, but they have a particular restriction, a spatiotemporal restriction, because the production of these compounds is highly demanding in energy. So the cells needs to know when to produce these compounds, because once the cell produces the compounds, there's no way back, right? So whether it accumulates pigments, aromas, or antioxidant compounds, or any other type of molecule, they need to restrict to a particular condition. So which are those conditions? Particular developmental cues, biotic stress or abiotic stress conditions, herbivory, etc. Just to give you some examples, aromas that are produced when the flower opens, pigments that are produced when the fruits start to ripe and accumulate sugars, carotenoids that start to accumulate in roots after parasitism of other plants, or in response to a pathogen, or even polyketides or alkaloids in response to herbivory. So we try to understand how all of these compounds that are produced in these particular regions are activated, and how these pathways are regulated by transcription factors. So which is the challenge here? We have plants, many different plants. These are non-model plants. Some of them are crops, some of them are not crops, and are quite difficult to cultivate, whether it's in greenhouses, in vitro conditions, or so on. In addition to this hard cultivation of these species, some of them do not have genomes with high quality, or have not been sequenced at all, or are really badly annotated. Or for some of them, we don't have much transcriptomic data, not even public or private, and also the metadata of this transcriptomic data is very poorly annotated. So the solutions that systems biology offer in this sense is we can apply different methods and different approaches to try to integrate as much as possible the omics data that is available. Or we can predict using computational modeling, gene regulatory networks and metabolic pathways. And also we can start applying metadata curation and classification algorithms, especially now that we are in this new era of artificial intelligence and language learning models. So one of the things that we do in terms of these solutions with systems biology is using network theory. And it's quite easy to study the regulation of metabolism through network theory, because the regulation of metabolism occurs through regulatory networks. So what is a regulatory network? The basic unit, as you all know, is a transcription factor binding to a cis element and regulating a particular gene that belongs to a pathway, a structural gene. But this basic unit aggruppates or is even more hierarchized in motifs. So you have one TF regulating several genes or elements, or a cross-regulation, or a hierarchical regulation. And these motifs go into one more layer of hierarchy, into modules. and then into a final network. So we can study the properties of each one of these different layers, and we can extract information that will help us to identify new genes, new regulators. One of the things that we can do is to infer a gene regulatory network, in this case using a gene co-expression network. So we have been developing in the last 6 years methods to generate gene co-expression networks in plant species. And of course, in a gene co-expression network where the nodes are the genes and the edges are the co-expression relationships, not all of these relationships end up in regulation, right? But part of this network can be used to infer a regulatory network. So in this case, the nodes are the TFs, the arrows indicate regulating another node, which is the target, and the arrow is basically the binding and the regulation, right? So how can we use GCNs to infer a regulatory network? There are many examples that I want to highlight and show you today that have allowed us to demonstrate that these have enormous potential to regulate, to find regulators, especially in non-modeled systems where transformation techniques or more sophisticated methods are complicated because of the challenges that these species impose. One important thing about networks, whether they are regulatory or co-expression, is that they are they have a scale-free topology, meaning that very few nodes have high number of connections or high degree, and most of the nodes have less or very little amount of connections or low degree. And this property allows us to identify the ones that are located here as potential hubs or potential regulators. Also, the gene co-expression networks follow a principle which is guilty by association, meaning that if you find a TF highly co-expressed with another gene, there's a possibility that they belong to a particular pathway and then one regulates the other. So one of the things that we have been doing is to try to download all the public transcriptomic data that you can find in public databases, such as Sequence Read Archive database. And we developed new method to re-annotate the metadata, because really what you find there is horrible. People submit data, transcriptomic data, without even saying to what organ they belong to. So we have tried to develop method, or even they annotate the data in places where they're not supposed to. So we try to re-annotate them following a pipeline that is based on regular expressions. So words or part of words that you can find somewhere, and you give different levels of validation depending on where you find those data. But also, depending on the species, we follow suggestions by experts, so field experts working in different species, because we need their information to know exactly which are the organs that are different between different species. So we have been able to download and re-annotate data for all of these species quite a lot. For instance, for Arabidopsis, more than 30K runs. For grapevine, almost 11,000. For tomato, 15,000 already. And once we are able to reclassify the public data for all of these species, we are now able to generate co-expression networks from the different tissues that we are able to reclassify, or from the whole data. So that is what we call a condition-independent network, or condition or tissue-dependent network. There are different methods. I'm not going to go in detail on the different methods, but we are following the aggregated approach, in which we generate a co-expression matrix from each experiment. This allows us to give the same weight to the different experiments. Let's imagine that there are 300 experiments in grapevine related to fruits. So each experiment, whether they have 6 libraries or runs, or it has 12 or 10, they all have the same weight. And we generate a matrix for each one of these experiments. These normalized count matrices are then transferred into Pearson coefficient, correlation coefficient matrices, high reciprocal run matrices, and finally we come up with a co-occurrence matrix. So every time 2 genes are co-expressed on an experiment, that is a 1. and you end up summing all the experiments. So you have a co-occurrence matrix with all the situations, all the matrices in which two genes from all the genome are co-expressed. So we visualize these networks, and then we extract information from these networks, right? As I already mentioned, we can extract parameters such as betweenness, which would be the case of this node, high connectivity or high degree, in the case of this one, in degree centrality, and so on. There are many, many mathematical parameters that we can extract from this and try to assert functions to the ones that have a lot of or a high amount of connections. So I'm going to give you some examples. So what is our study model? It's ripening. So we are interested on how non-climacteric fruits accumulate metabolites, but also on how they program or trigger other metabolic pathways such as sugar accumulation and so on. They are quite well-established models for non-climacteric fruit ripening. This is the case of strawberry and grapes. And this is a very nice, actually, representation of similarities between these two systems that was generated in the lab of David, actually. And as you can see, ABA in the two species seems to be a quite conserved hormone, accumulated at ripening, and actually it has been demonstrated that ABA is one of the main hormones related to ripening in non-climacteric fruits. However, there are other crosstalk with other hormones, particularly brasinosteroids at the beginning of ripening, but also other hormones such as gibberellins or jasmonates. So in the case of grape, in the last 20 years, there have been the molecular characterization of many of these transcription factors. If you see here, from fruit set to start of ripening to ripening or over-ripening, there are many TFs that are expressed in different stages of the development of the berry. All of these genes have been characterized through molecular standard approaches, so a few targets have been identified for them. What we have been trying to do is to identify the whole group of targets these genes have, or all the cystroms. And how we do that? First, we integrate or we overlap different data sources. One of that is the gene co-expression network, so we use it as evidence. But this co-expression evidence, we also overlap with cystrom data, basically all the regions on a genome where a TF can bind and potentially regulate. Such is the case of the results deriving from DAPSEq or ChIP-seq methods, and also the transcriptomics of plants that either overexpress or are mutant or knockout for a particular TF. The overlapping of these data sets allowed us to identify putative targets, high confident targets, or very high confident targets. And actually, we've seen in the last, let's say, 15 years, with all the amount of ChIP-seq data in model species, that TFs are able to bind in many regions of a genome. But not all of those regions are translated in changes in expression, in regulation. So there are many theories about that. People think that TFs basically are sitting in the genome and traveling all around until they found the specific environment, meaning other TFs, meaning open chromatin states, and so on. And then is when they regulate. So binding, although it's a proxy for regulation, is not necessarily meaning regulation, right? So we need to overlap binding with other data sources to try to identify putative regulators and putative targets, which those need to be then validated by molecular approaches. This is the different type of potential targets that we identify, and this legend is what I'm going to use in the following slides, so just for you to understand. This would be a high confidence target or a very high confidence target. This is a repressed target, a putative target, because it's only bound in gene co-expression data and systrom data, or only bound genes, which are those that are only found here. This is an example of the different transcription factors in grapevine that we have been characterizing by using this approach. As you can see, the number of bound genes is huge, from 3,000 to 10,000 genes being bound by these different transcription factors, but only a percentage of that is reflected as potential targets. Actually, if you see here, between 20 and 60% of a GCN, that would be all of this circle here, is validated by DAPSEC data. Okay, so each data set on its own is valuable, but it's not enough. We need to integrate and overlap these. Okay, so one example comes from the regulation of a particular metabolic pathway, which is the stilbenoid pathway. The stilbenes is a branch of the phenylpropanoid pathway that competes with flavonoids, and it's found in only some species in the plant kingdom, actually non-related species, so it's a very nice case of convergent evolution. Stilbenoid syntases, which are the first committed step of the pathway, are polyketide type 3 enzymes, just like chalcone syntases. A few mutations on a chalcone synthase gives the activity of an STS. And this mutation seems to have occurred in evolution in plants independently. So we have very unrelated plants producing stilbenes. Grapevines are one of them. So these in grape are very important because the main compound that is an stilbenoid is resveratrol. I'm pretty sure you all know, have heard of resveratrol. Anti-aging, antioxidant, and many other fabulouses that this compound gives. But it's very interesting from the regulatory point of view because it's only found in some species. So is the regulation of this pathway also specific? Actually, we demonstrated in this work that subgroup 2 MIP transcription factors, which in Arabidopsis regulate other processes, are involved in grapevine as regulators of stilbenoid synthase. This was the work of Luis while he was doing his PhD in the lab. So basically, DAPSEC data allowed us to identify all the bound genes. Most of the bound genes were in promoter regions. We found the consensus and so on. But what was interesting was that most of the binding, in the case of the stilbenoid synthases, was very close to the transcriptional start site. Each line here corresponds to one particular STS gene. There are like 40 genes in the genome. And when you compare that to a group of random housekeeping genes, you see that there is an enrichment very close to the transcriptional start site. In some cases, you have a second binding site, so you can see this right here in this histogram. And this allowed us to confirm that at least this gene, MIP14, that belongs to subgroup 2, was binding to basically all the family. And this was quite interesting because before it had been described as a regulator of stilbenoid synthase, but there was only one STS confirmed. So this is again a different way to represent the overlap between GCNs and DAPSEC. Here, this is all the genes that are co-expressed with either MIP14 or MIP15, which are these two genes belonging to the subgroup. And on the top part of the network, all of the genes that are validated by DAPSEC. Many of these genes belong to the stilbenoid pathway, so they are STS, but there are also other like hydroxylases that are potentially related to the pathway, lacases that are known also for the dimerization of these compounds, and also some transcription factors, such as worky transcription factors. And since then, this has allowed us to formulate a lot of hypotheses, demonstrate regulation by doing molecular approaches, or interaction of these genes with worky transcription factors, and so on. One of the things that we did was to overexpress these transcription factors in grapevine plantlets transiently And we see here the temporal transient overexpression of this gene, MIP15. We confirm the production of the glycosylated version of resveratrol, which is PSAID. And then we did transcriptomics of these plants. So we did transcriptomics, microarray in the different time points. This is just a selection of genes that are co-expressed, are bound, and that we also find them differentially expressed. So this is, let's say, the list of very high confidence targets that we know that are not only bound and co-expressed, but also activated with the transient overexpression of this gene in grapevine. What is interesting is that when we analyze this group of genes, and you see them reflected here, these are all the different branches of the phenylpropanoid pathway, plus the shikimate pathway, which is the one producing phenylalanine, the main precursor of the phenylpropanoid pathway. We see that this gene is binding and regulating the shikimate pathway genes, and the early phenylpropanoid pathway genes, and the stilbenoid synthase. So each color means that it's either regulated by one of the two MIPs or both of them. Gray means not bound, not regulated, and you can see that there is an enrichment of the early stage of the pathway. So this gene regulates the shikimate pathway. There's a lot of phenylpropanoid being produced, phenylalanine being produced. This enters the phenylpropanoid pathway. There's an activation of the first enzymes of that pathway, and then the stilbenoid synthase and all of these enzymes that are related to the derivatives of stilbenoids. Just to close up in a different way, so these two genes are able to regulate the primary metabolic pathway related to the phenylpropanoid pathway by producing more phenylalanine, and then the early pathway and the stilbenoid branch. This is how we knew the pathways were regulated before starting to implement these approaches. So, for instance, MIP14 and MIP15 was known to regulate this gene, one gene on the family. We also had MIP transcription factor that it was known to regulate the anthocyanin pathway, or one involved in the regulation of the proanthocyanidin pathway. However, when we apply these integrative approaches, overlapping different sources of omics data, we come up with different additional regulations. So all of these additional regulations, we can identify them by integrating these methods, right? So, for instance, we know now that this group and this group not only regulate their particular pathways or branches, they also regulate the early phenylpropanoid pathway, which is logical. If I want to produce a metabolite, I want to stimulate the whole pathway, even the first enzymes. Another very nice case is the characterization of a gene that we identify as a regulatory, as a main or key element in a regulatory network of volatiles, in particular terpenoid compounds. And we identified these in Chen's PhD thesis by studying natural variation, basically a variegating berry, which was able to accumulate pigments only in some stripes of the berry. What we did was to do different omic approaches to characterize the differences between this skin section and this skin section. As you can see, there is a complete depletion of production of anthocyanin in these regions, so we don't find any anthocyanins at all. So we dissected this area and this, and we compared them with transcriptomics. One of the interesting things was that, of course, the white skin section was highly enriched in genes that were more expressed in relation to photosynthesis, carotenoids, and other isoprenoids such as terpenes. So this was quite a surprise for us, but all the other processes were somehow expected. Also, it was expected that, of course, the pigmented section of the berry was highly enriched in genes related to the phenylpropanoid pathway. One of the genes that was induced in the white skin that had not been characterized so far was this transcription factor, MIP24. In model species, MIP24 is related to flower development. However, we saw that it had high expression in the late stages of ripening, so it was potentially related to the late ripening and over-ripening. And we saw that it was induced in these stages, more induced in the white skin compared to the red skin. So we said, okay, let's try to implement these approaches that we had already implemented for other transcription factors to try to understand what is the role of this TF. So this is, that went fast. Are you doing something and you want me to finish earlier, right? Give me a second. I don't know why this went so fast anyway. So one of the things that we did was, of course, to do DAPSEC and gene co-expression networks. But first, the gene co-expression network allowed us to identify a high connection of this TF with terpene synthase genes. The terpene synthase gene in family is quite big. There are more than 80 genes. Here you see them represented. All of these are terpene synthase genes. And this is MIP24. The gray edges correspond to co-expression relationships. So we see that there is a high co-expression of this family with this transcription factor. And actually the edges that are colored in red are the ones that are confirmed by DAPSEC, that not only are co-expressed but also are bound. So we hypothesized that this transcription factor could be potentially regulating the expression of these terpene synthases. So what we did was to do the identification of the regions where this MIP transcription factor could be regulating the terpenes. Here, just like with the stilbenoid synthase, you see different TPS genes that are known to be bound by the transcription factor, and there is also an enrichment very close to the transcriptional start site. We focused on a couple of them. One of them was this monoterpene synthase that produces beta-osimen, which is the terpene synthase 35. There is an enrichment of binding very close to the TSS. So we were able to amplify this promoter, fuse it to luciferase, and do dual luciferase assays. We saw that MIP24 was capable of activating the expression of this promoter. So we found luciferase activity only when we found, when we could infiltrate another transcription factor, which is MIC2, that we knew from model plant system that was an interactor of this type of this subgroup actually of transcription factors. So we said, let's try to find the homologue of the one that had been characterized in Arabidopsis, which is MIC5, and we identified it in grape, and we used it, and we demonstrated that only when these two are present, one or the other, together with MIP24, the activation of the TPS promoter is activated. I'm not going to go in detail on this work, but we characterize other targets and other processes, and we end up realizing that MIP24 is in charge of controlling the production of terpenes, monoterpenes primarily, but also carotenoids, such as lutein or beta-carotene. And all of these molecules, we postulated that they were related to photoprotection. So what we think is that in the white skin section of the variegated mutant, there is an increase of terpenes. I didn't show it here, but we actually quantified terpenes, and there are more terpenes in the white skin compared to the red skin. Also carotenoids. And we believe that this increase in these compounds in the white skin compared to the red skin is in a way to protect the cells, the epidermal cells, from the radiation, and in which, in the case of the pigmented skin section, you already have the anthocyanins, which are perfect sunscreens for protecting from radiation. So it's a way to bypass the lack of anthocyanins. So more or less, that was the model that we generated. Okay, so these are two examples of many transcription factors that we've been able to characterize in the group. However, is there a central regulator controlling these transcription factors? Do we have the regulator of the regulator? That is something that we were very interested. So we try to look for different transcription factors that could be regulating all of these TFs, so orchestrators, but also in the case of ripening or over-ripening, because we know that these ones are activated at the beginning of ripening, but MIG-24 is activated at the end of ripening. So one of the main candidates that we obtained was NAC60. NAC60 is actually one of the potential orthologs of RIF. That is one of the genes that the group of David is working with in strawberry here. So there was a similarity in sequence, and we actually saw that this NAC60 is a gene that is one of the first ripening biomarkers that is expressed just before all the other biomarkers of ripening. So we have here in green or in gray different markers like pigment, cell wall related enzymes, or genes related with different processes related with ripening, like sugar transport and so on. And you see that NAC60 is expressed just a little bit before. So this allowed us to postulate that this gene could be related to basically the regulation of all of the processes of ripening. Once again, we did what you already know. We did GCNs of fruit-specific GCNs. We did transient overexpression in grapevine, and we did microarray data, and of course we did DAPSEC. Here there is a representation of the position of different binding sites, but also regarding with the overexpression of the NAC transcription factor in grapevine. So the y-axis is the expression of this bound gene, and the x-axis is the position compared to the transcriptional start site. Many genes that are bound are related to hormones that actually are known to be involved in ripening, such as ABA, these ones here, or other processes like auxins as well, or even jasmonates. And one interesting thing was that we found among its bound and regulated genes, we found MI14, the first TF that I showed you, and MIB1, which is another TF that we characterize that is related to the regulation of the anthocyanin pathway. We demonstrated the activation of the MI14 promoter using a luciferase assay. These are 3 independent DAPSEC libraries where we show that there is binding, whether we use leaf or berry genomic DNA, more or less in the same position. So this enrichment corresponds to binding in this section around 1.5 kb. We used amplification of this promoter fused to luciferase, and we see that NAC60 regulates positively the expression of the MIB14 transcription factor. So it's a regulator of the regulator of the stilbenoid pathway. We check the same for other processes, such as the degreening pathway. So stay green is a gene that is related to the degradation of chlorophyll that is activated just before the beginning of ripening. And MIB1, which is the regulator of anthocyanins, of the red pigments. There is binding not only of NAC60, which is this one, very close to the TSS, but also of other 2 NACs that we had been characterizing also with DAPSEC at the time, which is NAC3 and NAC33. All of them bind very close to each other in the promoter of the stay green promoter or in the promoter of the MIB1 promoter. We actually demonstrated that the co-infiltration of these NACs, so the combinatory infiltration of these NACs, could activate very highly the promoter of these genes. So for instance, NAC33 with NAC3 or NAC33 with NAC60 were high inducers, the same here of these promoters. So this allowed us to, so the analysis of DAPSEC data allowed us to generate hypotheses about potential interactions of these NACs. And actually we did BIFC in grapevine protoplasts, and we demonstrated that these NACs are interacting. So probably they are binding to the same elements, and they are interacting, and they are regulating the activity of these promoters, and when they are together, they are increasing the activation. Now, when we look more in detail the binding genes and the putative targets, we not only identify the transcription factors of specialized metabolism as regulated by this NAC gene, we also found primary metabolic pathways being regulated. And this was very interesting because it had not been really identified in grapevine any type of dual regulator, both primary and secondary metabolism. So NAC60 regulates, for instance, aromatic amino acid synthesis, so chorismate, phenylalanine, and so on. Other amino acid-related enzymes, but especially sugar-related metabolism. So transporters, syntases, and many different enzymes that are known to be related to the import of sugar in fruits, or to the transformation of those sugars. So for instance, invertases that are in charge of breaking down sucrose when it enters the fruit and so on. And now we are trying to demonstrate whether these are real targets. We know at least that they are high confident targets because all of these genes from primary metabolic pathways or secondary metabolic pathways are bound, are co-expressed, or are regulated by the overexpression of these genes in transient conditions. Then, by the way, how am I with time? I think we are okay. This is like in the congresses. If you see that I'm talking too much, you just put the red light. So what we did was try also to identify if these TFs being regulated by NACs could have a feedback loop and could have cross-regulation. So one of the things that we did, as we had been characterizing MYC24 and MYC2, we did a very simple transcriptomic experiment of the co-infiltration of these two genes in grapevine. So we did transcriptomic of the infiltration or the transient overexpression of these two genes. Here simply we show the empty vector compared to the double transformation or the single transformation. So the biggest separation is when you co-infiltrate with the two TFs, as you would expect from the data that I showed you before regarding the DPS activation. And we identify very interestingly that NAC60 and another NAC such as NAC11 were among the overexpressed genes in the transient activation of MYC24 and MYC2. So could it be cross-regulation, right? So this is simply a way to represent the binding. So here is the binding because we did the DAPSEC of MYC2 as well. So this is the binding of MYC2 comparing to the TSS. This is the binding of MYC24 compared to the TSS. And these are all peaks, bound peaks in bound genes, right? So for instance, you see here NAC11 being bound by both MYC24 and MYC2 in a very similar region. So this region for MYC2 and this region for MYC24. So probably they are interacting, binding together in the same region and activating the activation of NAC11. We also found NAC3 in these two spots here, NAC60, also MYC24, so there could be auto-regulation. And we tested all of these potential interactions here, again with luciferase assays. And we see that, for instance, MYC24 with MYC2 activate the promoter of NAC11 in the same way as NAC60 or NAC61, which is another TF that I'm not going to talk about that we also worked on, that is related to over-ripening, also regulates. So we looked for transcriptomic data, previously published transcriptomic data, to try to understand how was the system in terms of time scale, right? So we know that NACs regulate MIBs. In this case, MIBs could also regulate NACs, but how are they sparsed in time? So we analyze different temporal series, in this case in 2 different cultivars, Cabernet Sauvignon and Pinot Noir, in different years, and we see a gradient of expression, a transient or a sequential expression of all of these transcription factors that helped us come up with a model that we want to test, in which NAC60 is the main regulator of, or the onset of ripening, but it's also involved in over-ripening, such as in the same way as NAC61, which we already know is related to over-ripening processes. MIG2 has a high expression just before Verson. We know that it regulates NAC60 because of the previous luciferase assays, so we think it has also a dual role, activating NAC60 here, but then joining MIG24 for activating these NACs, also regulating terpenes, regulating other processes, and we are right now testing this at the molecular level. Just to finish, we are not not happy enough by characterizing those few NACs, but we are trying to characterize the whole NAC family. This is the co-expression network of the whole NAC family, and we are interested in this group here in red, which are very specific NACs. So we did DAPSEC of all of these NACs, here representing that all of them are mostly binding genes in the very proximal promoter regions, so very close to the transcriptional start site. Actually, many of the bound genes of all of these very specific NACs are very shared, conserved. So what we think is that there is a redundancy. There must be a redundancy between all of these NACs. There are some specific pathways that are potentially regulated by each one, but we think that they are mostly binding and regulating the same genes. So this redundancy could allow us to identify not only one regulator of ripening, but the whole cluster of NACs. And this is new data that I have been looking for, thanks to the work of David and Carmen. Basically, we have been trying to understand whether what we find in grape is similar to what they have found in other species, like strawberry. So we did an orthology analysis between bound genes of NAC60 and bound genes of RIF, which is the TF that they work on. There is a lot of shared orthologs among the bound genes between strawberry and grapevine. So looking at these just briefly, without doing any characterization, but just looking at those briefly, we found many interesting things that are shared. For instance, ABA biosynthesis or signaling related genes, such as these ones, or transcription factors that respond to ABA, sugar metabolism, also anthocyanins. So these are shared between these potential orthologs. There are other processes that could be specific. For instance, NAC60 regulates stilbenoids, which are not present in strawberry or other plants. And at the same time, maybe RIF could be regulating other things that NAC60 does not do, right? So I guess that we can look at that in detail together. And with that said, I just want to briefly comment of one of the most proud things that we have generated in the lab, because it's a fair platform in which we offer resources for exploring all of this data that I have showed you, not only in grapevine, but in other species. We have been able to download public data, reprocess, reanalyze, and offer different tools for the communities. So gene co-expression networks, gene catalogs, co-expression data, expression heat maps, DAPSEC data, and so on. Of course, the amount of data, available data for each species is different. We're working with David and Carmen in strawberry as well, with other rosacea species, with microalgae and so on, and we would like to increase this platform with more species. So if you're interested, please let me know. What are the things that we offer in this platform, which is called PlantaeVis, for Plantae Visualization Platform? We offer this type of tools. So we offer different apps that you can explore, navigate through. For instance, gene cards, by inspecting literature and expression data that is available Publicly, we generate these gene catalogs or gene cards in which you find all the information that we can gather for a particular gene, including references, phylogenetic analysis, co-expression data. So here you can see each point is one run from an SRA experiment that is found in the public database, so you can inspect the expression on each one of the tissues. You can go and, with the cursor, touch one and see which run, which experiment corresponds to. We have developed this for Arabidopsis, for cannabis, for grapevine, for mulberry, for tomato, and other species. You can also see gene expression atlases. In this case, for instance, this is an hydric stress atlas where we can see the expression of a list of genes of interest and how they behave in different experiments. This is basically a meta-analysis, right? We have here different experiments that are available in the databases, so you can compare how the genes behave in one experiment compared to another. We also have a genome browser for DAPSEC data. We have this for tomato and grapevine so far, where you can see binding regions or peaks close to genes in the genome. We also have developed a tool that is basically the integration of transcriptomic and metabolomic data. It's called the TransMetaDB, and you can see for different experiments how genes correlate with different metabolic compounds, and there you can again formulate hypotheses of potential pathways being regulated by transcription factors and so on. And related to the co-expression networks, that is one of the things that I commented about. We have different tools. The app is called the Aggregated GCN app, and we have different tabs or tools within that app. One of them is you can put the name of a gene, any gene of interest in any species, and you will get a list of the 400 or 300 most co-expressed genes with that gene of interest. And in that set of co-expressed genes, you can do on-flight analysis, such as, for instance, GCA, so gene ontology analysis. In this case, this is the gene ontology analysis of the genes that are mostly co-expressed with MIP14. This is also navigable, so you can go with the cursor, touch this Manhattan plot, and here you will see which term is enriched. You can download this as a Excel file as well, because we want to be fair. Also, you can have in this other tab, you can give a list of genes and see how those genes interact among them. So this is just an example. We gave a list of stilbenoid genes in orange, MIP15 and MIP14 that we knew that were regulators. Other genes related to proanthocyanidin or anthocyanin, and this is the output of the tool. It shows you clearly that there are different pathways highly co-expressed with the regulators, a little bit separated from the rest. And you can do this with any list of interest. You can also explore how similar is the co-expression that you get from one tissue from another. So this is values of co-expression in the leaf network, values of co-expression in the co-independent network, values of expression in the berry network, and you can play around and see if the genes are highly co-expressed and are shared in one network compared to another. And that's it. I hope that I didn't go too much on detail. If you still need any information on the platform or any of the things that we do, please give me an email. And these are the people that I would like to acknowledge for each one of the different things that I showed you today. Thank you. 10 minutes late.
Thanks a lot for this talk. It was very clear and inspiring talk. During your talk, I saw some notes that resemble a lot the defense response of the plant. So how, now I'm wondering, what is the difference using the same nodes, MIC2, the MIP24, you have a completely different response in a leaf that is actually decrease the quality tissue. And in fruits, you have the opposite.
Yeah.
And to attract the herbivores and enrich the tissue.
Yeah, the tissue is, starting from DAPSEC, we have been doing DAPSEC and comparing DAPSEC performed on genomic DNA extracted from different tissues. We see some differences, but most of the bound genes are there. So the tissue from which you extract the genomic DNA that you challenge to ATF is not very much affected. It could be affected mostly based on methylation, because methylation changes binding, and you have different methylation patterns of DNA depending on the organ. But we don't see much. But what gives the specificity on a tissue is the expression more than the binding. So whether for instance, the NAC are expressed in other tissues in addition to berry. So we know that, for instance, the NACs participate in leaf senescence. So leaf senescence activates a lot of processes that are somehow common to berries in the sense of metabolic pathways. So metabolic pathways that could be related with, on one side, defense, but on other side, attraction of seed dispersers or attractants of other type. And so what we think is that transcription factors such as NAC60 could have a particular role in leaf, which is more related to senescence, or pathogen attack. So for instance, we know that NAC60 is activated in response to botrytis infection in leaves, or other pathogens. So it could be promoting the production of stilbenes through the MIP14 gene, because stilbenes are antipathogenic compounds. So resveratrol, the reason why grapes produce resveratrol is mostly because it's a phytoalexin. It has an impact on fungi. But it also has another very nice effect, that when we drink the wine or we eat the fruit, it gives health properties to us, right? So this gene turns to activate different programs, but seems to be depending on different inputs and tissues. So yeah, definitely we see overlap of processes related to pathogens, such as that one.
The final result is completely different.
Yeah. So that's why it's so important to overlap DAPSEC data with expression data, for instance, the GCNs. If we have done, for example, for MIP14, we have compared what happens when you overlap DAPSEC data with leaf GCN or with berry GCNs. You have many shared genes, but you have specific genes. So there are maybe targets that are specific to leaf, targets that are specific to berry. So that's one of the nice things that you can obtain by doing these condition or tissue-dependent co-expression networks.
Thank you.
Nice talk, thank you. In your analysis, especially when you consider co-expression data, you mainly look, if I get it right, to positive regulation activators. What about negative
Very good question.
regulators?
That is a handicap of our method. Our method only can extract positive correlation so far. There are other methods that are well established for extracting negative correlations. Our method so far works very well for positive regulators, but we have not been able to do it for repressors of pathways. But there are methods, so yeah.
Anti-co-expression, so like opposite.
No.
When one is on, the other one is off. And the other
Yeah,
one
I mean, the method we have not tried for anti-correlation. So what we do when we generate these high reciprocal ranking matrices, we keep only the highest co-expressions. But we could do it also with the lowest correlations. We have not tried it. It could be implemented. It could be implemented, yeah.
I think would be
Yeah,
useful.
definitely. I mean, we know that many transcription factors not only are positive or negative. Some of them are positive and negative, depending on the context, the interactors, and so on. So definitely that would be something additional to do in the following, the following years.
Looking forward.
Thank you.
Any other question? Yep. Sorry, maybe I lose something. How are you evaluating the gene expression? I didn't get.
The co-expression, you mean?
The gene expression.
Of what? Of what?
The genes that are expressed in the tissues. How you are evaluating that?
Oh, yeah,
The
definitely. Yeah. I mean, we are, depending on the gene, we are looking for whether these genes are specifically expressed on a tissue. Extracting the information from the GCNs also allows you to know if that gene is expressed in that tissue or not. Because when you do the networks, we use filters that if the gene is not expressed above a particular amount of reads, we take it out of the network. So if let's say in grapevine you have 47,000 genes, the networks will not have that amount of genes, only those that pass a filter. I don't know if that answered the question.
Not at all. Sorry.
But can you be more specific?
Yes, if you are using mRNA, or what are you using for watching in the tissue or in the cells, what genes are expressed?
So RNA-seq data, that is public. So SRA is the database.
Are you capturing? You are capturing mRNA or
Yeah,
proteins?
of course.
Okay.
Yeah, you were referring to that. Okay, yeah.
Yeah, sorry.
Yeah, so we use SRA, and SRA is only sequencing data.
Okay.
NGS, Illumina data.
Yeah, and you said that the database SRA is not complete. The SRA database that you are using.
Yeah.
I saw that the Plant Atlas ORG has a very good database. Maybe it's interesting for you.
Which one?
Plant Atlas.
Plant Atlas.
Plant Atlas organization.
Okay.
International universities that are.
But it's a database for raw data?
I think so. I never use it. But
Okay,
I
I'll
attend
inspect
from time
it.
to time to the seminar that they are organizing.
Okay.
and
SRA
looks like
is the most common database for uploading sequencing data. The point is that journals, when you publish your work, they always ask you for submission in a public repository. There are many public repositories. You have SRA, which is international. You have ENA in Europe. You have Chinese databases in China that are also very good. But they are not connected. Well, ENA and SRA are connected, but still the way in which people submit data is really poor. Because people just want an accession ID to put it in the paper and say, this was submitted to with this ID. But they don't care about describing well the experiment, so you can reuse that data later on. That's a big problem. So I think that computational biologists, we are moving to a new stage now, in which we are trying to implement AI models to predict the organ or the tissue or the condition based on the expression. So that is something that I know that many people are moving forward on that, because we cannot trust in researchers annotating their data. They don't want to annotate their data. Whether it's because of jealousy, because they don't have time, because this is just a protocol for your paper to be published. Unfortunately, people do not annotate their metadata. And that is not only bad for the community that could reuse the data, but also for you, for yourself, right? Because you're going to reuse the transcriptomic data always, always.
Okay, thank you.
Yeah.
Very nice talk.
Thank you.
The variegated grapes.
Yep.
Are they natural?
Yeah, they are natural. This is a very infrequent phenotype. It's found in 5% of the berries on a vineyard. It's a French variety. It's called Viquignol Noir. And has been quite stable for the last 60 years. On each season, you find variegation, but it's only in some berries. only in some berries.
Now, have you seen those berries or no?
Yeah, I have
Yeah,
dissected them myself.
I know. Okay. Have you noticed, because you are working with mathematics and, you know, maybe you are not working with them. Have you noticed differences in pruning on the two parts of the grape?
That's a very good question. I don't think that anybody has analyzed that because it's not a commercial vineyard. It's a collection garden, a variety garden. So there's only like 6 plants in a research institute in Bordeaux, where they found that and they kept that mutation. But nobody has done really experimentation on the plants.
Right.
Yeah.
It's because the terpenes.
Mm-hmm.
Because one of the most, important components of prunes are triterpenes. Is the acid, or lanoic, or solic, right?
Mm-hmm.
It's like amyrins, but is the acid form, right? And this is important, because they got a photoprotection value as well, and also it's very.
Are these triterpenes?
Yeah, they are pentacyclic triterpenes.
Okay. Mm-hmm.
Yeah.
We didn't measure triterpenes, but what you say has a lot of sense because among those terpene synthases, many of them are triterpene synthases.
Yeah.
Yeah.
I imagine because more than about 30% of prune are triterpenes.
Okay.
It's important. Okay, just a suggestion.
What I do know is that in other species, this gene has been characterized, the MIP24, and it's known to regulate triterpenes, and they have been characterized. So I'm pretty sure that in grape could be the same.
In the peels of the fruits, there is a negative correlation between flavonoids, anthocyanins, and waxes.
Waxes.
Yeah.
Because we found that anti-correlation between anthocyanins and terpenes or carotenoids. But with waxes, it could be interesting.
Waxes in general. Waxes are a mixture of terpenes, long-chain alcohols, and acids. So that's the importance of that.
Okay. Yeah, that could be interesting to follow up. Yeah.
Okay.
Tom,
I think there was
just talking about this, is it known why this variegation phenotype is something really
different
expression
the
of that?
The phenotype is because MIB1, which is the gene that regulates anthocyanins, in the white varieties, it's knocked out because of a retrotransposon inserted in the 5' UTR. So the gene is not expressed. And it cannot regulate anything downstream, so the anthocyanin pathway is completely blocked. What we see in the variegated berry, we did molecular marker analysis and sequencing, and we see that the white skin behaves like a white variety, and the red skin behaves like a red variety. The original variety is red, so it's functional alleles of the MIB gene. Somehow, we don't know how, or either a cell that had the heterozygous allele invades other tissues like L1 and L2 tissues and become all that area white, or there is a jump of the retrotransposon again in the gene blocking in some particular cells, and those cells propagate and become but it's very unstable, so it's not always the same amount of variegation. You have sometimes stripes, you have sometimes half of a berry is white, half of the berry is red, so it's very heterogeneous.
Hi, very nice talk. Maybe my question is very naive, but it's more on the technical side. I was wondering, when you integrate different transcriptomic data from different experiments and different samples and everything, do you do a normalization step specific to each data set and then you integrate them, or do you integrate and
you normally apply batch analysis, so because they come from different experiments, sequencing platforms, the data on itself is different. So if you plot the expression data on a PCA, all the expression data of SRA, even data coming from the same type of experiment, for instance, control and botrytis infected berries. If you plot them in a PCA, I'm pretty sure they will first clusterize by experiment rather than control. And but you need to do batch effect analysis. Yeah.
Okay, thank you.
Thank you very much for your talk. Very nice. About this last comment that you have about the variegation, this grape that really fascinates all of us today. It really sounds familiar to something that is called position effect variegation in Drosophila I. So my question is, if you see this gene, this transcription factor, is it close, you know, in the localization in the genome, is it close to centromere, telomere, or heterochromatic regions?
The MIVE1 gene, which is the one responsible for the phenotype, it's located in the short arm of chromosome 2, but I don't know if it's close to the centromere.
So if you check that in Drosophila, I can give you the data, is dependence on the boundary of heterochromatin, which change or can slightly change its mitosis.
Okay.
This means that the gene can be silenced or on off, and one is like silenced because heterochromatin is spread towards the gene and is divided by mitosis. Then this patch of cells is on or off, depending on the heterochromatin state. So maybe that is something that could be explaining what you have.
That's interesting. What we have done, in fact, has been to do layer-specific analysis. So we have done molecular markers in L1 derived and L1 plus L2 derived tissue, and we see differences there as well. This allowed us to at least hypothesize that this was a periclinal chimera in which cells from one layer was going to another layer. We cannot demonstrate it, but that is what we think. So we think that the mutation was on a particular layer within some group of cells, and those cells were able to move to another layer. So this is known that it can occur depending on the division. It divides in a different plane compared to the rest of the neighbors. And one of them goes and moves into another layer. And we think that when it moves to the other layer and it spreads, now you get mutation both in L1 and L2, and then you get the white phenotype, the white. But that could be another possibility as well.
Okay, thank you.
Anyone else? No? Okay, thank you very much.
Thank you.
Mexican sky, drink some margaritas by a string of blue lights, listen to the mariachi play at midnight.